Okay. Or not?
Yes.
Are you seeing my screen now? Okay.
Yes.
Okay, so as Alba said, I'm from Poland and I'm working in two places. One is International Center for Translational Eye Research, which is located in Warsaw. And another is Nicolaus Copernicus University. It's Faculty of Physics, Astronomy and Informatics, and it is located in Toruń. And from 10 years, my main topic of my research is two-photon vision. So I would like to share my research on this topic, part of research on this topic with you. So the presentation outline is at the beginning, I will explain the two-photon vision mechanism, and then I will focus on spectral sensitivity for two-photon vision. Also, I will show you what is one of the difference between two-photon vision and one-photon vision is the quadratic dependence of brightness on power, and the consequences of this for psychometric function of two-photon vision and for also the clinical application of two-photon vision, which is two-photon microperimetry. At the end of the talk, if it wouldn't be too long, I will also show you some results about pupil light reflex, which is caused by two-photon stimulus, and I summarize my talk at the end. So, two-photon vision rely on visual perception of pulsed infrared beams as having color ascribed to half of the wavelength. So it basically means that when we use pulsed infrared laser of 1000 nanometers and we focus it on our retina, we will not see red light, we will see green light. So you can see here that this is like another range of visible range of electromagnetic radiation, which is from 800 to 1300 of nanometers, and it is the second place when all the rainbow, perceived rainbow of colors is present, and this is the range when two-photon vision occurs. And the phenomenon was first observed in 1965 by Russian physicists, which were working with gas laser neon hydrogen laser. However, it remained unexplained until 2014. And the explanatory mechanism was provided by a large interdisciplinary team of researchers, of which I was a member. And I was responsible in this team for the psychophysical experiments. And one of psychophysical experiments on humans, one of the results of these experiments is shown here. And it rely on presenting, so we are using the system which was similar to scanning laser ophthalmoscope. And in this system, we presented two vertical lines on the retina to the participants, and one line was emitted from the infrared tunable laser of 200 femtosecond pulses and 76 megahertz, repetition frequency. And second line was visible, normal visible light from the CW source, which was simply halogen bulb lamp, but we filter it by using monochromator, and the filtering was adjustable for the participant. So the subject task was to match both colors by changing the wavelength of this visible light. And what we found, we performed this experiment for 30 healthy participants, and we found that they all can perceive that colors, and that colors are very close to double frequency line perceived by this process. However, it was not exactly double frequency light. There were a little bit reddish than this double frequency light. And on this plot, you see the results of all similar experiments that were reported so far in the literature. So our is showed by these red points, and you can see here that this visual perception of two-photon stimuli is very common. So you can obtain it with using similar lasers in every laboratory. However, all that colors, almost all of them, are slightly shifted to the red from the double frequency line. The one only exception from this red shift is these results, but these are very old results from 1979 from Dmitriev, and in their experiments, the Stiles-Crawford effect was not fully controlled, probably. So it is not confirmed by the others. And the reason of this shift to the red part of the spectrum is still unknown. Okay, so what is the mechanism of this two-photon vision? So for normal one-photon vision, the vision starts from the absorption of photon by visual pigment. But where this visual pigment is located? So we've got retina. Retina is composed of several layers of neural cells. And at the bottom of the retina, you've got photoreceptors. And there are two kinds of photoreceptors in mammal retina. This is rods and cones. And in these photoreceptors, also at the bottom of them, so at the bottom of the retina, there are place which is called region, which is called outer segment. And these outer segments is composed of discs or foldings, in case of cone, of cell membrane. And in this cell membrane, there are highly packed opsins. And opsin is, here you've got rhodopsin, is composed of protein plus visual pigment. And this visual pigment in opsins of rods and cones is 11-cis retinal. And this is the guy that absorbs the photon. So when you've got normal vision, you've got this 11-cis retinal, it absorbs the photon and it changes its conformation to all-strands retinal. And this is the first step of phototransduction, which is as you probably know, the mechanism of conversion of photons to electrical signal, which then could be used and transmitted by other neurons of our visual pathway. And what is the difference to two-photon vision? So everything goes basically in the same way. However, this absorption at the beginning, it is not the physical process of one-photon absorption, but the physical process of two-photon absorption. And two-photon absorption is process that occurs when a molecule or atom interacts with high photon fluxes. When you've got high photon fluxes, it may happen that two photons will be interacting with molecule in the same moment, and then they will be simultaneously absorbed. And then a molecule goes from ground state to excited energy level, but this change corresponds to the sum of energy of both photons. It means that basically by using this process, you can use infrared light to excite molecule to the energy level that under one photon absorption can be excited by visible or even ultraviolet light. Sorry. But I said that this process is not very likely. So you need high photon flux. And high photon flux means that power of the beam is very high. And when the beam power is very high, it will result in a lot of energy deposited in the tissue. So why we are not destroy the retina with these lasers? So we are not destroying the retina with these lasers because most of the time the laser does not emit any photons. Here you can see the example of calculation for the beam that we have been using at the beginning. So the experiment that I show you. So 1000 nanometer, 200 femtosecond pulse width and 76 megahertz repetition rate. So the 76 megahertz repetition rate means that the time between pulses is 13 nanoseconds, and pulse width is 200 femtoseconds. So maybe it is not very easy to understand the difference between these two. However, when we recalculate it into the distances by using simple physical equation, you could see the difference, because the separation in space between these two pulses would be like almost 4 meters, and the position of the width of the pulse would be only 60 micrometers. So you can see here that most of the time the laser does not emit any photons. And so intervals between pulses are long enough to cool the tissue and prevent to damage it. So two-photon absorption occurs only during pulses, and photoreceptors are very sensitive to light. And even single absorption events can be converted into visual perception, what we know. So this is the reason that two-photon vision can occur. So these two things, the proper design of the pulses and the high sensitivity of photoreceptors to absorption of the absorption events. And two-photon absorption is commonly used in, for example, in two-photon microscopy. So we know what is the equation that links number of absorption of isomerizations that occurs on visual chromophores with parameters, with global parameters of laser and optical systems. And this equation also describes the process of two-photon vision. And as you can see, one important thing is that the number of these absorption events, which converts into the brightness, the luminance of perceived luminance of the stimuli, depends on the square of average power of the laser. And I will focus on this talk on this kind of difference between one and two photon perception. However, I would like to say now here that also this number of two photon absorption events also depends on time of pulse width and on repetition frequency. So by changing these parameters of the laser, you can also change the brightness of the stimuli, of two-photon stimuli, by, for example, lowering the repetition rate with the same pulse width, or by lowering the pulse width. You can also change the luminance of two-photon stimuli. Also, here you've got very high dependence on the numerical aperture. And this is also important for two-photon vision because, as you probably know, the numerical aperture of the eye, even when you've got fully dilated, when you are using fully dilated pupil, is not very high. However, the practical numerical aperture is even lower because of high optical aberration. You cannot have the well-focused light at the retina level, even when you are using this fully numerical aperture of the eye, which is 0.23. So to modify the perception of the two-photon vision, you could also play with the optical aberration to increase the practical numerical aperture of the system. Yes. However, these two things will be not not the subject of my lecture today. Okay, so what is the two-photon spectral sensitivity? So probably you are aware that light above visible range is not impossible to perceive. Perception depends on light intensity. And we know, for example, that when you are using 1000 nanometer light, it could be perceived. However, the visibility threshold would be 10 orders of magnitude higher than in the maximum, yes, of sensitivity, which is in the fovea center. It is like 555 or 56 nanometers. However, this perception, this long end perception close to 1 photon normal vision is connected with the perception of red color. And we perform experiment with our lasers. We measured the threshold, the visual threshold of 3 participants at the fovea center. And we measure when we are using the 800 nanometer laser beam, the beam was perceived as red, and the visual threshold lies the relative visual threshold lies in the perfectly in the line, in the literal line. And when we increase the wavelength, it also follows the known dependence of known process. However, when we are starting to go into the longer wavelength, like 850 and 160 and so on, the perception slightly changed. The perception of color slightly changed because the beam was perceived not as fully red. It was like red with addition of blue or violet color. So we can measure basically 2 visibility thresholds. One higher threshold was connected with the perception of this additional blue color, and lower threshold was connected with perception of the red color. And when we plot these thresholds, we found that this connected with perception of red color matched very well to the known curve from literature. However, these blue ones were not. And when we increase the wavelength again up to 950, the difference between these 2 thresholds became lower and lower. And then we just couldn't measure 2 thresholds because the beam was perceived only as blue, without any red addition. And then the perception was like blue, green, yellow, and up to red. And the visual thresholds for that beam was deviates from the normal 1 photon spectral sensitivity of the human retina. So I would like to say that green points indicate this 2 photon spectral sensitivity, and it clearly deviates from normal 1 photon visual sensitivity. And to explain the shape of this sensitivity, we do some operations on the normal 1 photon V lambda curve. And these operations were like follows. First, we divide this normal V lambda curve by the transmission of ocular lens and macular pigment in the visible range. Then we double the wavelength, and then we multiply this doubled wavelength hypothetical V lambda by the transmission of ocular media in near region. And then we obtain that curve, and you can see here that it coincides quite well with our data, and the only region of exception is in this blue part of spectrum, this common blue part, and it might be connected with with this the presence of these 2 thresholds in this region. And another reason would be the thing that macular pigment level is very individual, is very different between individuals, and we are using the standard values from the standards. However, we are we are like 3 different people, so our participants could have not very medium or mean level of this macular pigment. Okay, so to conclude, the absolute thresholds which were connected with the perception of red color in the range 750 and 950 fit very well classical data for one-photon vision. And the visual thresholds for two-photon vision have been measured in the 800 to 3000 nanometer range, and these thresholds were connected with the perception of color, which is close to half of the wavelength of the stimulating laser. And there is small range when there is an overlap between these two mechanisms. And the maximum of two-photon sensitivity lies between 1050 and 1100, and it coincides very well with transmission window of water of ocular media. And the overall shape of the sensitivity curve, two-photon sensitivity curve, resembles the photopic luminosity curve with wavelength multiplied by 2 and corrected by the ice transmission in both regions. And the next part will be devoted to quadratic dependence of perceived luminance on power. And I will show you the results of 3 experiments, which shows that this quadratic dependence exists. First experiment was performed on 3 healthy volunteers, and we project them 1 and 2 photon stimuli. Both were perceived as green because it was 1045 nanometer and 522 on the green background of increasing intensity. And the intensity of this background increased from 0 to 1000 and 800 trollans. And we asked them to measure both thresholds for this 1 photon and 2 photon stimuli in the same background, and then plotted the results on the log-log plots. And we expected that if it would be quadratic dependence, the slope of this plot should be around 2. And because this is log-log plot, yeah? So yes, it was. As you can see, for 2 of them was very close to 2, 1 was 1.7. So these results indicating the nonlinear character of two-photon vision. This experiment was a little bit different because it was without the background, and we present the rings, 2 rings for our participant at the same time. One ring was 1040 nanometer and the second ring was 520. And we also asked them to match the intensity of this 520 nanometer beam to the intensity of infrared beam to have the same or very similar brightness of both. And then when we plot the results, also on log-log scale, we found that that slope was 1.7. This is not exactly 2. However, it is very close to it. And why it is not exactly 2, we are not very sure. However, we think that it may be connected by the fact that the color of 1040 was not exactly the same like 520. As I said to you at the beginning, it is more yellow. So maybe it will change the matching, the brightness a little bit. Similar experiment was performed with these 2 beams, but with different stimuli. We projected here letters of different sizes of human retina and also at the same time, and asked them to make the same matching with background off and background on. Background was white in this case. And here we found that mean slope was even a little bit higher than 2. Yeah. So we've got this quadratic dependence on brightness on power, and what would be the consequences of that? One of consequences is different shape of psychometric function for two-photon vision. Psychometric function describes the detection probability of stimuli of known intensity. So basically, you've got when you plot the stimulus intensity in the log scale, and when you will be plotting the probability of detection of this, you could have some region which defines going from close to 0 to close to 1. And you can define here, for example, threshold level as the 50% of detection, of probability of detection. However, when you will be using lower intensity of the stimuli, the probability of detection would not be 0, but close to 0. And for higher, it will be not exactly 1, but close to 1. So the parameters that describe the shape of psychometric function are connected with this experiment, with the probability of detection, and the one which is important for us in the context of this change between 1 and 2-photon vision is beta, and beta, it is the slope, and it defines how narrow is this function. And when we've got quadratic dependence of brightness on power, it affects the slope mainly. Because the slope would be, because here we've got logarithm of stimulus intensity. So basically, theoretically, the psychometric function would be twice narrower on this scale. So it means that the distribution of possible thresholds, which is connected with this, so when you are measuring thresholds, you not obtain always the same value, which is exactly connected with this, but you obtain some distribution of thresholds in this region. Yes, so this distribution should be twice narrower, and then it means that the accuracy of visual threshold determination would be higher for that kind of process. And we tried to show it, and we performed several experiments. Here you've got the results of 2 of them. One is published, and this is results for 1 person only, and here are results for 3 different persons which perform many trials of this psychometric function measurements. And what we find that exactly the beta, this slope for 2-photon psychometric function is significantly higher for 2-photon vision in comparison to 1-photon vision. And the ratio of these slopes was 1.65 for these 3 subjects. Here, this is the example for only 1 subject, and it is exactly 2. And the consequence of this smaller or narrower psychometric function can be shown on the repeatability of the measurements of visual thresholds. So we measured the visual thresholds for the cohort of subjects. Here there were like 17 subjects by using 2 kinds of methods, method of adjustment and staircase, adaptive staircase 4-to-1, with 1-photon and 2-photon stimuli. And what is important here, you can see that this region is almost exactly twice narrower for 2-photon stimulus in comparison to 1-photon stimulus. So here is very similar results from older paper. Here is not exactly 2, however, it was 1.1 decibel for infrared and 1.7 for vis. So what is the consequence? The psychometric function is significantly narrower, which is caused by quadratic dependence of brightness on power, and it makes two-photon vision a more precise tool for testing the visual threshold, and it leads us to an application of two-photon vision. So the application which is mainly tested at the moment is two-photon microperimetry. And what is perimetry and microperimetry? Maybe you are aware or maybe not. If you are, it depends on your background. However, it is also called visual field examination, and there are devices which are called perimeters when they are using such big ball, or microperimeters when they've got functionality of imaging the retina at the same time. And the examination relies on the measuring or testing visual sensitivity at a different part of the retina. For example, you can use the grids like here and test the visual sensitivity of the retina in different position, and then compare it with normal visual sensitivity and say if the photoreceptors at this place are functioning normally or not. And the test is important mainly for glaucoma progression and also for diseases which affects photoreceptors, for example, AMD. Sorry, I've got some problem with, oh, okay, sorry. And we perform such kind of test with our device for cataract patients. Why we are focusing on cataract? Because we was looking for the main, how to say, main difference between 1 and 2 photon stimulation, and we thought that, for example, when you've got cataract, the absorption of visible beam would be higher in this lens than of infrared beam. So we could use two-photon microperimetry for testing the visual sensitivity of the retina of patients with cataracts. And it was very hard experiment, because we basically have to go to clinic with our laboratory system. We went there 3 times, and it was during pandemic, because we started it just before the pandemic in these 2 sessions, and then we have to wait like half a year, because we would like to measure their sensitivity before and after cataract surgery. Yes, so we have to wait a little bit to have them have all operations performed, and also the all eye healed and so on. And the second visit was just in the middle of pandemic. So. But we managed to measure almost 30 people before and after. Then you've got whole procedure, and we also use Maya commercial microperimeter to have the comparison to regular microperimetry, and our system performed exam with 1040 nanometer beam. No, not 40. It was 1028. Because we were using fiber laser, 1028 nanometer laser beam and 520 visible beam. And when we compare the sensitivity difference before and after, so we measure this sensitivity before and after, so we can calculate the sensitivity difference for visible beam and for infrared beam, we found that, in fact, the sensitivity for infrared beam was less changed after cataract surgery than for visible beam. And also we checked that the correlation between these 2 sensitivities difference were pretty high in our subjects. So we think that this confirms our hypothesis. Yes, the sensitivity for visible beam changed more after cataract surgery than for infrared 2-photon beam. However, We also performed the Maya exam, and it was very, for us, it was very strange that the sensitivity measured by Maya was also very small change. The change was similar like for infrared beam. However, there is no correlation between our system and Maya at all. So we think that the changes, the difference between these two systems, our and Maya, was too large to compare the results between them. For example, Maya uses broadband white LED to project the stimuli, and we have been using lasers. So probably this broadband white LED is not affected such in the same way like for example 500 like green beam, yes, by cataract. The second difference, what was caused by the cataract surgery, was not only the change of transmission of the lens, but also, of course, change of refraction error. Because people before have their natural lens, so they've got very the very different refraction error in this group. And after that, they were corrected, yes, because the intraocular lens correcting the refraction error as far as it is possible. And when we check if this change of refraction error correlates with the sensitivity difference, we found that for Maya it does not, but for our system it does, and it does significantly for infrared. So it probably means that correction of refraction by our system was subjective, and probably the patients was not fully corrected, and it affects the results for infrared. However, it is even better for our hypothesis because it means that this slight change of the slight improvement of the visual sensitivity in infrared two-photon beam maybe is connected with the improvement of refraction, yes? So if we would remove this factor, maybe the change wouldn't be for infrared beam would be even smaller. So to conclude this part, the cataract surgery affected more one-photon than two-photon sensitivity, which is due to less scattering and absorption of 1028 light in the cataract lens than for green light. And the refraction change caused by intraocular lens implantation also influenced the two-photon sensitivity of tested patients. And further experiments are needed to separate the impact of both factors with objective control of refraction correction. There are more data on two-photon microperimetry in clinic because, for example, the most of the papers are published by first author Grzegorz Łabus, which I cooperate with because there is one system which is constantly present. It is in Heidelberg University Hospital, and they measured by two-photon microperimetry many patients with many diseases. And if you are interested, I will. These are the papers. And at the end, maybe I've got several minutes to also present something slightly connected with the others, which is pupillary light reflex for two-photon stimuli. We also measure it because we would be interested how, because it is something which could also characterize the response of visual system for the light, and we would like to we would like to know if this response is similar or not similar for 2-photon stimuli in comparison to 1-photon stimuli. So we made these experiments with using two kinds of laser stimuli, which were projecting the spiral on the central fovea, and one was 1040 nanometer, and the second was 520 nanometer beam. And also for comparison, we would be using the LED stimuli of the same wavelength like the visible laser, which would be like fully, it would be projecting fully, not the spiral, but the circle. And we found that the reaction of pupil for 2-photon visual stimulus is significantly weaker than for both visible stimuli. And for both visible stimuli, the reaction was similar. However, it was slightly weaker for the laser stimuli than for LED stimuli. And also we focused on the parameters, several parameters that could characterize this reaction, like latency time, time of maximal constriction, mean constriction velocity, minimum pupil diameter, and so on. And we found that almost all of these parameters were significantly different for 2-photon stimuli and 1-photon stimuli, and one only which was not different was latency time. So we think that observed weaker reaction of the pupil for 2-photon stimulus might be caused by weaker stimulation of rods by 2-photon stimulus and more negligible scattering in the eye media, because stimulation occurs only in the focal region and fewer photoreceptors are stimulated. And also, I would like to say that we think that cones are probably more susceptible for 2-photon stimulation than rods because of two other results, which I will shortly present here. One was dark adaptation curves, which were measured for 2-photon and 1-photon stimulation, and we found that the difference between cone plateau and rod plateau was 1 order of magnitude smaller for 2-photon stimulation than for 1-photon stimulation. And also, when we compare the exact shape of sensitivity map for 1 and 2-photon stimuli, with it was scotopic sensitivity map. So it means that there is a difference between central part of fovea when you've got only cones and the other part of fovea when rods are present. And the difference between visibles, for the visible stimuli between these two parts was 6 dBs. However, for the 2-photon stimuli was only 2 dBs. So this is another observation that confirms that maybe the difference between cones and rods in 2-photon vision is not so high like in 1-photon vision. So to summarize, the 2-photon vision occurs for wavelengths in the range 800 and 1300 nanometers, reproducing all 1-photon colors, and the maximum of 2-photon sensitivity lies between 1050 and 1100 nanometers and coincides with transmission window of water. And the quadratic dependence of brightness on power means that the visual threshold can be determined with better accuracy, so the 2-photon vision should be useful for 2-photon microperimetry. And several facts might indicate that cones are more susceptible for 2-photon stimulation than rods. The smaller difference in scotopic sensitivity between the fovea and perifovea center, the shape of the dark adaptation curve, and the weaker pupil response, and also this systematic redshift in perceived color also could be explained by the difference in levels of stimulation of different kind of photoreceptors. So two-photon vision is a novel and interesting phenomenon in vision sciences, and there are many aspects, as you can see, of this phenomenon to investigate in future. And here I would like to thank my younger colleagues, which are Agnieszka Zielińska, Marcin Marzejon, Oliwia Kaczkoś, Juliusz Solasz-Niesłuchowski, and Mateusz Grochowski, who is not present in this picture, and also my other collaborators, which are depicted here. And I, of course, would like to thank my funding agencies of my studies. So thank you very much. And I would like to answer